Yeah, please go ahead. Yeah.
Okay, thank you. Hello everyone. I am Adeyemi Oluwayomi. Nice to be here. Thanks for having me. I'm here to discuss a paper we recently published in veterinary parasitology on exploring the genetic diversity of Eimeria acervulina, a polymerase chain reaction restriction fragment length polymorphism PCR-RFLP approach. Starting with the background, yes, as to what Eimeria parasites are and why they are important. Eimeria parasites are protozoans of the phylum Apicomplexa. They belong to the coccidia group, and these coccidians are developed in the intestinal mucosa or submucosa of diverse vertebrates, where they cause tissue damage, they initiate enteritis and malabsorption. In the poultry industry, is considered the most important parasite. It causes coccidiosis, especially under intensive conditions, and both subclinical and clinical forms of the disease are economically important. And the global poultry industry has recorded losses up to 10 billion pounds every year. The etiology of Eimeria, they have a direct life cycle. They are transmitted fecal-orally. Birds, susceptible chickens become infected when they ingest food or water that is contaminated with oocysts, the infective stage of the parasites. And these parasites, Eimeria, are highly host-specific. Species that infect a particular host do not naturally infect another kind. And in the chicken, where Eimeria seems to be the most important, economically important in chicken, about 7 different species are well known to infect the chicken. Eimeria tenella, or Eimeria necatrix, Eimeria acervulina. These species vary in pathogenicity, they vary in immunogenicity, and they vary in the sites, predilection sites within the chicken host. Recently, we discovered 3 new species were discovered recently: Eimeria lata, Eimeria nagambi, and Eimeria zaria. Yeah. The control of Eimeria parasites. Currently, Eimeria is being controlled by routine chemoprophylaxis using anticoccidial drugs, both synthetic and natural anticoccidial drugs, and like vaccines are also being used. Various formulations of like vaccines are being used. But currently, these methods, these tools are facing limitations. There's been emergence of drug resistance, consumer safety issues with the drugs also, and also the high cost of producing vaccines and limited production capacity for producing vaccines are limiting the current control tools. So in the literature is proposing subunit recombinant vaccines as better alternatives. They are cheap and easier to produce compared to the wild type vaccines or the live parasite vaccines. But we have some questions. Will Eimeria parasite develop resistance to these subunit recombinant vaccines if we plan to use them in the future? To answer that, we will need to know the extent of genetic and antigenetic diversity that is already existing in the field with Eimeria parasites. That brings us to genetic diversity studies, various studies that have been conducted to establish the genetic diversity of Eimeria. But most studies are focused on Eimeria tenella, possibly because it's the most important, most pathogenic and most common Eimeria parasite of chicken. So several studies have studied genetic diversity of Eimeria tenella, but there's been no study for the genetic diversity of Eimeria acervulina, especially multi-locus diversity studies. And why is this a problem? Because Eimeria acervulina, though not as pathogenic as Eimeria tenella, is also highly prevalent globally and is the most important cause of malabsorptive coccidiosis, which is also associated with significant economic losses and is a major threat to the welfare of chickens. Different molecular genetic tools have been proposed or have been used rather to study the genetic diversity of Eimeria parasites. PCR-RFLP is one important tool that detects genetic polymorphisms located within recognition sites for restriction endonucleases. Why is PCR-RFLP coming up on board now? Because they are reliable, sensitive, and affordable, especially for low to medium and low to middle income countries, where that makes comparative analysis between different regions very possible. It has been applied to study the genetic diversity of Eimeria tenella in the UK and Irish chickens, as shown. So this study aimed to therefore develop and apply a robust, accessible, and cost-effective multi-locus PCR-RFLP approach to differentiate Eimeria acervulina field isolates. We also try to explore the extent of genetic diversity that exists within and between African and European Eimeria acervulina populations to determine the extent of diversity and to see, if eventually the subunit vaccines that are being proposed to be used in the future will last long. A PCR-RFLP marker was developed, following about 9 steps. 9 steps are listed on the slide. The first was to identify coding sequences, gene sequences of Eimeria acervulina in GenBank. These sequences were then the homologous sequences within the outing strain of Eimeria acervulina genome were identified also for primer design using nucleotide blasts in ToxoDP. Forward and reverse primers targeting each locus were designed, and that was used and then the primer specificity of this, the primer specificity of the design primers was also carried out, was assessed. Primers were then used to amplify the target loci using reference strains of the outing reference strain of Eimeria acervulina and the Weybridge reference strains. The product of the PCR was sequenced, and locus was selected, and a locus was selected as an RFLP marker if it had a restriction endonuclease recognition site that is disrupted by either the outing strain or the Weybridge strain, if it exists as a single copy per genome, and if it's suitable for primer design. The final steps was then to go on to carry out the enzyme enzyme restriction digestion. And the PCR applicants from the target sites for the reference strains were subjected to restriction endonuclease digestion, and the digested products were resolved by agarose gel electrophoresis. So all of these 10 steps, I beg your pardon, constituted the marker development. These markers that were developed were then applied to field samples that were collected, about 40 field samples of Eimeria that were positive for Eimeria acervulina. Fecal samples positive for Eimeria acervulina were collected from Nigeria, Egypt, United Kingdom, and other European countries, as shown in this slide. And similar protocols in steps 5, 8, 9, and 10, as described for the reference strains, were applied to these field samples. To analyze the data, we identified the genotype of each SNP as based on the correspondence with the outing and the Weybridge reference strains, where there occurred a single band after the digestion and the electrophoresis, where a single band occurred indicated that a SNP type was associated with restriction site disruption, and where there were 2 or 3 bands that complicated the maintenance of the restriction site. And in some cases, we found two both band profiles suggesting that such samples had pluriconal population infections, and the brightest band was taken as the most dominant genotype. The genotypes were compiled on Microsoft Excel and were used to create SNP haplotypes for each genotype. The genotypes for each of the markers were then compiled together for each sample to create an haplotype for that sample. And the haplotypes that were derived were combined with equivalent data and were compared. Findings. We found that only 9 markers out of the plethora of markers, about 13 target loci that were tested, only 9 markers met the selection criteria. Markers are listed in table 1. Most of these markers are surface antigen proteins of the Eimeria parasites. And the table also shows the fragment patterns, the band patterns of the different strains for outing strain and the outing strain on the left, the outing strain on the left, and the Webern strain on the right. We found that for Nigeria, not for the samples that were collected from Nigeria, not all the samples amplified successfully at each of the target markers. These are the markers that were assessed in the field samples. And you can see from the table that about only 17 for IMP1, for immune-marked protein 1, I beg your pardon, only 17 samples amplified successfully. And none of the samples amplified for MZ92 130 protein, for example. Only 5 amplified for actin depolymerizing protein. But for samples that were collected from Egypt and other parts of Europe, all the target loci were successfully amplified. The fact that some samples from Nigeria were not the markers were not amplified successfully in some of the samples collected from Nigeria is probably indicating the presence of some extent of polymorphism in the primer binding site. So it's possible in the genetic sequences of the samples collected from Nigeria, there were some variations in the primer binding sites that did not allow the primers anneal successfully. Table 3 shows the summary of the PCR-RFLP genotyping for all the isolates of Eimeria acervulina that were collected from Africa and Europe. Because some markers did not amplify successfully in Nigeria, we were only able to come down with using 6 markers to assess genetic diversity. And as shown in table 3, out of 20 samples that were analyzed in Nigeria, 30 haplotypes were detected in them, and 14 of these haplotypes were distinct. So out of 20 samples, 14 distinct haplotypes of Eimeria acervulina was detected. And there was also occurrence of about exactly 50% pluriconal infections. And there were for other parts of Europe, 14 haplotypes were detected out of 7 samples. 10 of them were distinct haplotypes. 4 of them were country-specific, and there was also occurrence of pluriconal infections, indicating high genetic diversity or haplotype diversity for Eimeria acervulina in these regions. We went on to try to draw or to plot a median joining phylogenetic network. And these notes are shown here. The notes represents the haplotypes, the unique haplotypes that were found in each of the regions. and you can tell from the network a lot of haplotypes existing, and very few of them were shared between regions. Very few of them shared between regions, but a high diversity of haplotypes are shown. Finally, we went on to find out the discriminatory profile of these markers, and found that in Nigeria, the immune map protein 1 was highly discriminatory for Eimeria acervulina, followed by each shot protein 90. In other Europe, actin depolymerization protein was the most discriminatory, also followed by micronein protein 5 was also highly discriminatory in other parts of Europe. This table just summarizes and tells us that discriminatory markers for the different regions vary. So the discriminatory power of the markers vary by regions. And if local studies are to be conducted in these regions, markers are already available that can, at a goal, help discriminate between Eimeria acervulina in these regions. The conclusions of this study: the study revealed that Eimeria acervulina populations were highly polymorphic in Africa and Europe, with high occurrence of polynuclear infections. There was no region-specific occurrence of genetic diversity. Genetic diversity was the same statistically across all the regions. High genetic diversity, but was similar across all regions, unlike it was described previously for Eimeria tenella that had a continental, a regional divide, where in the north there was there was low diversity compared to the southern parts that were conducted in that study. And finally, high genetic diversity of Eimeria acervulina in this study indicates that genetic recombination might be very common in these parasites, between isolates of these parasites, with the potential to influence the longevity of the efficacy for future subunit vaccines. Thank you for listening. I'll be happy to receive your questions.
Yeah, thank you, Adiyemi. So now, before going for the questions, I invite the reviewer present their views.
Good morning from here. Good evening also, night, evening from different parts of the world. Thank you for your presentation, Mr. Adeyemi. I found the paper very, very interesting to read. If I had the opportunity of sharing it with you, you would have seen a lot of markings on the paper. So it's a very informative paper, a good job. I like the approach, which you employed in the study. But I have a few questions because I had a little bit of confusion in understanding especially the sampling, okay? Because when it comes to this type of work where you are trying to look at the population structure for the development of vaccine or for therapeutics, it's important to be able to, get properly what goes on with the sampling. So because the sampling is a very important factor at the end of the day, in looking at the genetic diversity that you're going to get. So the first question I would ask has to do with the sampling. Now, you said you sampled 40. No, first of all, the H and the W types of this Eimeria acervulina. Okay. Yes. So these two variants, what is specifically different about them?
Okay, thank you. There are reference strains of Eimeria acervulina. The strains were isolated, both were isolated in the UK. In the United Kingdom, yes.
So what is the genetic variance between them? Like, what's really the difference? Is it so much? And because yes, I noticed that you designed your primers okay, with more using the H variants.
Yes.
Yeah,
yes.
Without using the W variant. So what exactly is the difference between these two variants of the references, the reference genomes for the two samples? It is interesting because I would really like to know. That's going to inform my next observation.
Okay. Should I go ahead and answer that?
Yes, please.
Yes. So these are reference strains that were collected from the United Kingdom, from Alton and Weybridge. These are locations. They are based, they are named based on the locations. And they have some level of diversity, polymorphisms. A lot of SNPs have been found to exist between Alton and the Weybridge strains.
Okay, so that explains why, both strains, the reference strains for the UK. Now, I wanted to ask next, why didn't you use both strains? Oh, it's gone.
I'm here.
Oh, okay. So why didn't you use both strains, like to find the region of alignment between the markers that you selected when you were aligning the reference genome of the H and the W, and then to look for areas where you could find degenerate markers in the primers in the areas of the region of the genome where these genes are? Why did you just limit it to just the Alton strain?
Yes, thank you for that question. The whole point of that, in why we use the Alton strain from the word go, was to first of all locate or rather get to the location of these genes or coding sequences that we were proposing to test, to know where at all they are in the Eimeria acervulina genome assembly. And we use the Alton strain because the Alton strain is the only one that has been
the genome has been fully annotated.
Yes, fully annotated. And
okay,
yes,
I get it. So the Weybridge strain is still undergoing annotation. That's why you use the Alton. So I get it. Yes. All right, because the thing is, if you see why I asked the question that what is the main difference, what is the difference between them? There are two strains, the reference genome in a particular environment, then it might be easier to design degenerate primers,
if
that would be more informative if both of them were aligned in the primer design process. So this takes me to the sampling now. I'm a little bit confused about what you did here. I see that you sampled broilers and layers all across Europe, Egypt, and in the UK and Ireland, you sampled broilers. Now, when you got to Nigeria, you sampled broilers and layers. So first question would be, did you separate out your results with respect to the types in terms of the parasites you collected between broilers and layers? That is number one. Number two, we do know that, Nigeria has some kind of different geographical spread. So was there, in terms of looking at the Nigerian population of this particular parasite, did you look at it vis-a-vis where did you collect the samples? Is it like, did you collect across the geographical zones in terms of Nigeria? And did you look at, if you did you look at what kind of, the genetic diversity with respect to the Nigerian population? And then, the broilers could be male or female. Of course, layers are female. So did you collect the male? Did you separate in your sampling male vis-a-vis female and all those kind of things? So I would really like a little bit of more information about your sampling. Then with respect to the sampling still, so you actually isolated the parasite from the feces of the chicken, right?
Yes.
Okay. So, and then in the isolation, it seems, if you say 42, okay, I see that the two reference genomes were present. Yes. So it doesn't mean you isolated one parasite from each of the chicken, or of the chicken that you sampled, just one parasite?
No. Okay. Oh, sorry. Am I supposed to answer now? Sorry about that.
Yeah, so you see why I'm a little bit confused in terms of your sampling, because at the core of population structure and genetics diversity is the sampling. So I would like you to elaborate a little bit on your sampling.
Yes. Thank you very much.
Yes, thank you.
Okay. Would that be all?
No, but this is a very important question that will lead me
Okay.
into what I'm going to say next.
Okay, great. Thank you. So I hope it's safe to say this is more like a baseline or preliminary study to start off with, to use PCR-RFLP to tell the genetic diversity of this parasite. It's not as extensive as we would plan to do in the future. And so when sampling, we did not take into consideration the whether the reproduction types of the birds. They're all chickens, and they all get infected with Eimeria acervulina. Although, of course, we might find out that there are different genetic types for different production types. Yes, but we did not put that into consideration. I mean, we're blind to that and just took it as Eimeria acervulina in chicken generally. We wanted to assess genetic. So we did not go down to production type delineation or sex delineation. And yes, the samples were collected from from commercial poultry farms. The only thing we tried to ensure was that these farms, none of these farms are currently using vaccines for control, because the vaccines were developed from the outing strain, and we didn't want that interfering with the results. Yes, so we went to farms that don't use vaccines, which there are a lot of them actually, because of the cost of the high cost of these vaccines. Yes, and yes, so in Nigeria, like you asked for Nigeria, where you saw we collected 20 samples from farms. Lagos was what was representing Nigeria in this study. It was just Lagos we collected, but it was extensive. It was an extensive sampling in Lagos, because we collected from farms in Lagos East, farms in Lagos West, where most of the farms are domiciled, and that was random enough, and it was representative to a very large extent. Although I know that these are the limitations of the study, and we really hope for a more extensive and more robust sampling in the future. Based on what you're saying, that I'm taking up as recommendations.
So that is exactly where I'm narrowing down to, Adeyemi. So yeah, go on.
Yes, because you also asked the question about if it was one parasite, that's one individual, we isolated from these farms that we went to.
Yeah, from each chicken that you sampled.
Yes, okay. So I would quickly do a rundown of how we sampled. Yes, I believe that's well explained in the paper, but for the purpose of this meeting, the talk, the samples, we collected pooled fecal samples from the farms. So we visit a farm and go into a particular pen where chickens are either on the floor or in cages, and they're dropping their feces in a collection tray or wherever. So we collect fecal samples from random points in the pen and represent it as one for that whole pen
area.
Yes, for that open. So that's a sample. That's one sample. Now there will be millions of oocysts of Eimeria in this sample. So we extracted, isolated the millions of oocysts as much as we could recover based on by salt flotation method. We isolated those, purified them, and extracted DNA from what we had. So what you are seeing as a sample is a population.
So it's a population of the parasite in that particular environment.
Yes.
Okay, all right. So I get you now. So I have a very good picture of the sampling, which will translate into the explanation or interpretation of the results that you've done. So now, I wanted to start by suggesting that, since we already know that the sample size is a delimitation, because when we start looking at population diversity studies, when the sample size are usually small, there is always tendencies for you to have those kind of high haplotype diversity because you have a small sample size. Yeah. And now we're not just looking at the Nigeria as a whole, you extended it further into Europe, and you picked some samples around Europe and then in the UK. So then we also know that from experience in genetic diversity with respect to Africa, there is a high level of genetic diversity in Africa, which explains why I'm not surprised that you did not, most of the primers that you got, that you used, worked for all the samples in Europe, in UK, in Ireland, and even in Egypt. Because if you look at the geographical distributions, there's a high likelihood that they're going to be using some kind of the same sample of species of chickens around those areas. So because of the small sample size, I believe, because of the small sample size within Nigeria itself, the mean, and then the fact that, you know, there's a kind of genetic diversity that we usually find when you look at living organisms around Africa compared to the different parts of the world, I think it's why you have those high haplotype diversity. So if you now look a little bit inwards, okay, like if you were able to get a little bit of more samples, okay, to add into this study, I'm sure that that haplotype diversity index will definitely come down. Now, so because it's sort of like when you use the word haplotype, your haplotype means that you're talking about a set of DNA variations that are inherited together. So those sites tend not to be, you know, subjected to, you know, crossing over, genetic recombination events during meiosis 1. So when you're now having high haplotype diversity, you're saying that in those regions, there is actually a lot of genetic recombination going on, which defeats the purpose of selecting those regions for vaccine production. So I would want to suggest that maybe a different approach you could try in this kind of study. Because of that, I would want to suggest if you would try to use genome-wide association studies. So you can do a robust, you know, GWAS study using genotyping. And in this case, you know, you could pick representative genomes. Already, they've done a lot of work in UK. As you see now, they have the Orton and Weybridge genome as their reference genome. If that kind of a study could be done, okay, for you to identify a potential reference genome, it might be a little bit difficult, but you could, I mean, these are lower organisms, you could get reference genomes and then do a GWAS study. And that will give you a lot more SNPs. You will not be restricted to this particular SNPs alone, because SNPs are usually, GWAS is always very effective when you are looking at, you know, genetic variations that are associated with a particular phenotype of disease. So in the future, I would suggest that maybe you could employ a GWAS approach, okay, in the type of, you know, in this study. So it's going to give us a lot of more information, and then it will definitely reduce the haplotype diversity indices, and then it will also reveal other interesting proteins or markers, molecular markers that you could use as potential targets for the vaccine development that will cut across. Of course, you don't want genetic diversity too much, I mean, at all, most of the time when you are developing, you know, a vaccine that will work. So do you get me, sir?
Yes, I do. Do you want me to respond to a few of the points?
Yes, I would like me to hear your view about it.
Yes, thank you so much. I've noted these recommendations down, and I think this is something to look forward to doing. Yes, really, yeah, very true. And I was just going to say that, yes, because you raised a question about the fact that we might be going too far, or rather, it might not be correct at the moment to conclude that there's high genetic diversity because of the small sample size. But we found that why we moved on to do this for Eimeria acervulina was RFLP, PCR-RFLP was also used previously for Eimeria tenella. And it was comparable sample size. The sample size was also small because we just wanted to try that out in the, at first, just to see what we get. Since Eimeria tenella has been widely studied and the genes and the coding sequences were well established, we tried that using small samples also from different pockets or places across regions. And we found out that the genetic diversity was lower compared to what we got for Eimeria acervulina, and which has something to do with the biology of this species. You are very right that we might end up having to include more loci, like you doing the GWAS study, and find out that the haplotype diversity is not as high as we thought it was. But we will still not be shocked if we find out that it's still higher than what was obtainable for the other species, Eimeria tenula.
Yeah, well, that would still be sort of like a speculative thing again, because
yes,
the study that you described for this particular species, they both have different biology. So I mean, they are both from the same genera but different species. So that could account for the reason why the genetic diversity in that other species is not as much. But the good thing about doing GWAS is that, you know, when you do a little bit of more random sampling, when you have a large data set, okay, it helps you to see a global picture of what is happening across the genome of the organism. Irrespective of what you have looked for, it helps you to determine more robust markers, you know, that you could use as targets. And you to even uncover a lot of more interesting information. And this could be like a project that at the end of the day, you know, I don't know where it would take you. It could take you to big places. So, and I know that a lot of my colleagues who are into especially things like plant biodiversity, they usually like to start from that global picture of GWAS and then try to come down, because this approach of PCR-FLP eventually is going to be a bit expensive at the end of the day. How many loci can you really design primers for? How many loci can you really capture? But now you're using a particular reference genome to design primers that are not working for your own particular population. So if you're able to sort of like narrow down to a reference genome in a particular area, or some few genomes that you have, then you'll be able to see a global picture of what is going on across them. So like I said, it's a very good study. It's a very nice baseline research, just like you said, like a kind of pilot study. Is very good. So, and I applaud I mean, you and your team for the work you've done. So I also wanted to ask, lastly, before I leave the floor open for others. So when you did your PCR for optimization of primers, did you use gradient PCR?
Gradient PCR, no.
Okay,
no.
So you just stuck to that 60 degrees Celsius to use. So it could also mean that it is one of the previous approaches to use when you're doing primers like this is to try a gradient PCR approach. And then you could also try touchdown PCR. You look at a wide range of temperatures from one beginning to the other, and then you run it through touchdown. And if you maybe try gradient PCR or touchdown PCR, you will be surprised at what a few degrees in temperatures could do in terms of PCR. A particular primer can work very well at, say, 58 degrees Celsius, and then you'll be surprised at 57 what you'll be seeing. The bands will be so thin. So I would suggest that even those ones that you had to resort to sequencing for you to be able to figure out the genetic diversity, if you tried a gradient PCR approach, touchdown PCR approach for your optimization, maybe that could also give you a little bit of, more information, positive output towards it. So with that, I think I'll just step down and allow others ask their question. Thank you very much for the beautiful work.
Thank you. Thank you for your recommendations too.
Yeah. Thank you, Dr. Pradeep, for your wonderful comments, and as well as sharp understanding of the subject. So I now request, floor is open for the questions. So I now request any audience if they have any questions. So either you please raise your hand, or probably you can just type your question in the chat box. Yes. So I see a question in the chat box here. So it's by Dr. Salisu. She was asking, like, is the result a kind of expected when variation is high? And also another question is, what was the sample size of Tenella? So yes, yeah, it's open for you now. Yeah.
Thank you.
Yeah.
Thank you. The sample size for Eimeria tenella, okay, we didn't work with Eimeria tenella in this study. It's a previous study, yes, also done by the team I collaborated with on this, but the sample size was similar to what we had for the Eimeria acervulina study. Yes.
Okay. Thank you. So Professor Sharma has a question. So I request Professor Sharma to probably you can ask your question directly. Yeah.
Yeah, my question is that what was the method of selection of the samples? Like when you select some samples, so your country is very wide and you have selected multiple countries. So what was the criteria for selection of samples from different countries, whether it was geographically distributed or you had taken some random samples?
Sorry, can you come again, please? I beg your pardon.
So, probably I can explain the question to you, Ademi. So Professor Sharma was asking whether what sampling strategy have you followed, whether it is random or stratified or other kind of sampling, because since your study involves different geographical locations, so that's his question. I guess if I'm right here.
Oh, okay. Yes.
Yes,
Yes. Sorry about that, Professor Sharma. Yes. We sampled randomly. We sampled randomly in these regions. Yes.
Okay. So, any other questions from the audience? Okay, so if you don't have any further questions, I think we can wrap up the session. So, yes. So I thank Ademi for his presentation, wonderful presentation, and his study. I I thank him for making his time, for giving his talk here uh, in this podium. I also thank Dr. Fadip for her review on the subject as well as on the paper. So I now hand out the session to Dr. Aniyo, for the final, final words. Yeah. I think Dr. Aniyo. I think there is some problem with your audio.
Yeah, can you hear me now?
Yes, we can hear you.
Okay.
Yes, we can hear you.
So in the absence of no, I think we can round up the session.
Yes.
Dr. Fadipe, I want to appreciate you for your time. Dr. Ravi, thank you for coordinating. And Mr. Riemi, thank you very much for accepting our invitation. And to everyone, I want to say a very big thank you for coming, for joining this session. Looking forward to see you in December for the last session for this year. So thank you so much.
Yes.
And have a
Thank you all.
wonderful day. Yeah.
Yeah. Thank you all. Thank you very much. Thank you. Thank you.
Okay.